Hey guys, so for this video, we have been tasked with extracting DNA from common fruits and vegetables using nothing but household materials, in which I would also try to explain how these materials work in correspondence to said DNA extraction all throughout said process. So, without further ado, let's go. So, for the setup, here are the materials that I have used. First was dish soap. Next were 2 containers, my fruit of choice, which was a kiwi, some hot water, salt, a gauze, and cold isopropyl alcohol. So the first step in making our precipitating solution is the addition of 1 tablespoon of salt. For this experiment, salt is important because it helps remove the proteins bound to the fruit DNA. Moreover, it helps separate proteins from the DNA during precipitation upon the introduction of the cold isopropyl alcohol into the mixture. Next, we add in at least 10 drops of dishwashing liquid. Adding dishwashing liquid is important due to its content of sodium dodecyl sulfate. Sodium dodecyl sulfate is a compound responsible for lysis or breaking down of the proteins in both the cell membrane and nuclear envelope, exposing the chromosomes that contain the DNA. In addition to this, sodium dodecyl sulfate helps break down the histones and other DNA binding proteins by denaturation, thus unwinding the DNA. After this, we add warm water to the solution to further melt the cell and nuclear membranes, as well as to prevent the breakdown of DNA by certain enzymes present within the cell. Lastly, we add warm water to the mixture in order to enhance the effect of the dish soap that was added earlier. And now to complete the precipitating mixture, we thoroughly mix the ingredients together. Now with the precipitating solution ready, it's time to prepare the extract. In this part of the experiment, I chop open 1 piece of kiwi fruit in which I empty its contents in a separate container. Now I add in hot water to the container and consequently mush the fruit into a pulp. This is done in order to break the cell wall of the cell, thus allowing the cytoplasm to leak out. And now I add in 5 tablespoons of the precipitating solution into the mixture in order to begin lysis of the cells, and later on to allow for the precipitation of the DNA to take place. Here, with the aid of my grandmother, we sift the large mushed kiwi into the container to mush the larger pieces further, allowing the remaining extract to be obtained. And now, we filter in the crude extract into a separate container to remove large particles such as seeds and etc. For the last part of the experiment, we proceed to the precipitation phase in which cold isopropyl alcohol was added. Cold alcohol was used to make the DNA precipitate. It is important for the alcohol to be cold to increase the yield by preventing the breakdown of DNA. Moreover, alcohol was used since DNA was insoluble in alcohol, therefore allowing its separation from the aqueous solution below. As we wait for the mixture to settle, the white layer forming above the mixture is the DNA of the kiwi fruit itself. Its consistency is viscous because it has already clumped together thanks to the alcohol added earlier. And there we have it. The DNA is already visible and thus it could be deemed that this experiment was a success. I had a lot of fun doing this, and once again, this is Adrian Conrad Dange signing off. See ya!